This plant is Edelingera elatior, commonly known as torch ginger. It is a perennial herbaceous species belonging to the family Zingiberaceae. This family includes economically important plants such as ginger, turmeric, and cardamom. The species is native to tropical Southeast Asia and thrives in humid, high rainfall environments. This presentation examines its morphology, reproductive biology, physiology, and phytochemistry. Kingdom, plant, clade, angiosperms, clade, monocots, order, Zingiberales, family, Zingiberaceae, genus, Edelingera, species, Edelingera elatior. As a monocot, it exhibits parallel leaf venation and fibrous root systems. Like other members of Zingiberaceae, it develops underground rhizomes for vegetative propagation. Edelingera elatior is a rhizome. It's perennial. The visible leafy stems are pseudostems formed by tightly wrapped leaf sheaths. The inflorescence develops on a separate stalk emerging directly from the rhizome. The structure, often mistaken for a single flower, is a compact inflorescence, composed of overlapping sterile bracts, multiple true bisexual flowers, a central reproductive axis. The bracts are modified leaves that protect immature flowers and assist in pollinator attraction. The rhizome functions as a storage organ. It accumulates carbohydrates primarily in the form of starch. This energy reserve supports rapid shoot development and reproductive growth. Vegetative propagation occurs when rhizome segments generate new shoots. This clonal growth strategy increases population density and competitive advantage in tropical understory ecosystems. Rhizomate's growth also enhances resilience against environmental stress. The inflorescence is adapted for biotic pollination. Bright pigmentation functions as a visual signal for pollinators such as bees and nectar-feeding birds. The floral architecture protects nectar from dilution in high rainfall environments. Pollination is primarily cross-pollination, which increases genetic diversity. The stigma and anthers are positioned to maximize pollen transfer during pollinator contact. This structural optimization improves reproductive efficiency. As a tropical species, Edelingera elatior is adapted to high humidity, warm temperatures, diffuse forest light. Its broad leaves maximize light absorption under shaded canopy conditions. The elevated inflorescence ensures reproductive visibility above surrounding vegetation. This vertical separation between vegetative and reproductive structures reduces competition for pollinators. Like other Zingiberaceae species, Edelingera eladia produces bioactive secondary metabolites. Identified compounds include flavonoids, phenolic acids, essential oils, terpenoids. These compounds serve multiple ecological functions: antimicrobial defense, herbivore deterrence, oxidative stress protection. Some studies report antioxidant and antimicrobial properties in extracts of the flower and rhizome. These phytochemicals contribute to both ecological survival and culinary value. Edelingera eladia plays a role in tropical ecosystem dynamics by supporting pollinator populations, contributing to understory biodiversity, stabilizing soil through rhizome networks. Its dual function as an ecological and edible species highlights the link between plant defense chemistry and human use. Edelingera eladia represents a complex integration of morphological adaptation, clonal propagation, pollination optimization, and biochemical defense systems. This species illustrates how tropical plants evolve structural and chemical strategies to survive and reproduce efficiently.